Hello friends, today we are going to study the SYBSc paper 2 plant physiology, and in this we are studying the chapter 6, that is seed dormancy and germination. That is part 2 we are going to study. In the first part we had studied about seed dormancy. In the second part we are going to study about seed germination. Okay, I am Dr. Sangeeta Kulkarni from the Department of Botany, Radhabai Kale, Mahila Mahavidyalaya, Ahmednagar. So we are going to study about seed germination. I had told you that this picture depicts that when a seed is put in the soil and gets all favorable conditions, it starts imbibing water, and then it starts germinating into structures called as radicle and plumule. The radicle, it moves towards the ground and develops into the root, and the plumule, it is developed from the in between the cotyledons and grow into a shoot. Okay, so this is the process of germination of a seed when all the conditions are favorable. Now this is a dormant seed which is showing you this green colored structure as the embryo. Then there is a brown colored cover which is called as the seed coat, and inside this there is the storage called as endosperm. Usually the embryo, it survives on the stored food, that is in the form of endosperm earlier, and later on in the form of cotyledons it is getting the food. Now what is seed germination? According to the definition, emergence of embryo in the form of seedling from the seed is called as seed germination. It is the emergence of embryo in the form of seedling from the seed or sprouting of the seed. You must have seen that when you keep any seed in water, or if you keep in that seed near to the water conditions, then you will see that it imbibes water, and slowly, because of that, cell division takes place in the seed and it germinates into a seedling. Okay. Now how does the seed, what are the steps in seed germination? First, the dry seed, it imbibes water. Imbibe means it soaks water. That is called as imbibition process, due to which there is activation of seed enzyme system. Inside the seed, the enzymes which are already dry because water was not present, so water becomes a transporting medium, and these enzymes, they get activated, and after they get activated, there is activation of respiration and energy is released, okay, in the form of ATP. Now is stored in the form of ATP and lost in the form of heat. Metabolic changes occur in the seed, and therefore the seed starts germinating. Okay, what are the metabolic changes in the seed that takes place? The seeds undergo metabolic changes during the seed germination. The complex reserve food is broken down into simpler food. That means once the enzymes are activated, what do? They convert the complex food material into a simpler food material. So one by one we will study what are the different metabolism which are occurring in the seed during the process of germination. First one is carbohydrate metabolism. During the metabolic transformation, starch which is present inside the seed is broken down into glucose and sucrose. It is utilized by growing embryo. The alpha or beta enzyme amylase help in the process. Okay, for this is the enzyme alpha and beta which are helping to carry out the process of carbohydrate metabolism. That means initially the seed contains starch, but when water arrives there, this starch starts getting converted into glucose and into sucrose, which is utilized by the growing embryo for its own development. Okay. Now next is the protein metabolism, the second one. During the process of transformation, the storage proteins are converted into simpler amino acids. That means they are also stored. There are very complex proteins that are present. They may be having tertiary structures, may be having quaternary structures, and when they are in the vicinity of water, they start converting into their chains, they become loose, and they start converting into simpler amino acids which are utilized by the growing embryo. So once this conversion takes place, it helps the growing embryo to take up all this material. The peptidases and the hydrolases enzyme are involved in the process. During this process, these are the enzymes which help in carrying out this protein metabolism. Okay, or change in the protein. Next is the lipid metabolism. During the process of transformation, the fats and the lipids are converted to fatty acids and glycerol, which are utilized by the growing embryo. That means they are stored in the form of fats and lipids, but when this imbibition process takes place, there is activation of enzymes. These fats, they get dissolved into fatty acids and glycerol, and therefore they are utilized by the growing embryo. For this process, the enzyme lipases help in the conversion. Thus, due to availability of favorable conditions, the metabolic changes occur in the seed and it germinates. Basically, favorable condition means that it is mainly getting water. Once water arrives, automatically all the biochemical activities, they are properly channelized because of this process. Now, what is the role of phytohormones to improve seed germination? You know that phytohormones means these are hormones which are like auxins, cytokinins, gibberellic acids, ethylene and abscisic acid. These hormones, they are basically either they grow, help, all they are basically helping to regulate the growth in the plants. Some of them may act as enhancers and some of them may act as inhibitors. Therefore, they also help. These phytohormones also play an important role in improving the seed germination. That we will study in this part. The mechanism of seed dormancy and germination are controlled by various factors that are present around the seed. Okay, so one of the factors are plant hormones. As I told you, it is dependent on conditions like water availability, environmental, you can say stress are there, then dry conditions are there, then other factors are there, and one of the factor which is important is plant hormones produced by both plants and soil bacteria. Interaction between plant hormones and the plant genes affect the seed germination. There are plant genes that are activated in the presence of specific plant hormones. You know that all the genes are present in the DNA. It is believed that when that seeds comes in contact with the plant hormones, these DNA or the genes present on the DNA, they get activated to create signaling. And once they signal, the kind of product is formed which is useful for the growth and development of the seed. Okay, therefore we have written that the plant genes are activated in the presence of specific plant hormones. The hormonal signaling of indole acetic acid, IAA means indole acetic acid, and gibberellins play an important role in seed germination. That means these are already present in the plants and the genes are activating them. So once they are these phytospecific plant hormones, they affect, then they are going to activate indole acetic acid and gibberellins, and therefore they play an important role in seed germination. The phytohormones like abscisic acid, ethylene, gibberellins, auxins, cytokinins also have a role in seed dormancy and germination process. Okay, along with these there are others also. There is a group of gibberellins, there is a group of auxins which are present, and therefore they play an important role in both the process as dormancy and germination. Okay, the plant hormone gibberellins are considered to be necessary for seed germination. Okay, gibberellins break the seed dormancy and promote germination. This part is very important, that the plant hormone gibberellins are considered as enhancers. So these gibberellins, they promote the process of germination, as they were discovered in the rice or the you can say rice plants, where they had shown that the rice plants, their height was increased because this produces the hormone gibberellin from Gibberella fugicora. Okay, correct. Then the hormones ethylene, cytokinin and steroids, they promote seed germination. Even these hormones, ethylene which helps in ripening of the fruit, then cytokinin which helps in cell division, and steroids, they are helping to promote seed germination. So along with gibberellin, these are also important hormones. Embryo dormancy is characterized by high ABA, that is abscisic acid, and the gibberellic acid ratio, whereas they are responsible, whereas the seed germination has high abscisic acid sensitivity and low gibberellic acid sensitivity. Okay, so it is having these things are in seed germination and dormancy. It has been found out that the ratio of abscisic acid to gibberellic acid is important, which creates either dormancy or it creates the germination. Okay, abscisic acid is the only hormone known to induce and maintain seed dormancy. So if the amount of abscisic acid is increased very high, then automatically it is going to lead to seed dormancy. Okay. Seed vigor index. It is the quality parameter of the seed which determines its level of activity and performance during germination and seedling emergence. It is one of the parameters which is used to study the plant index, or you can say the seed index, in which it says that the activity and performance of the seed during germination and seedling emergence, at what rate it will grow, it is called as seed vigor index. Okay, seed vigor index is a parameter which is assessed to understand germination and viability tests of the seed lot in the field or in storage. That is, if you keep the seeds for very long time and then one day you try to grow them, you provided all the conditions, the rate at which it will grow. Some may grow very fast, some may grow very slow, some may grow when only specific conditions are given. That is called as seed vigor. Okay, at what rate it grows or it germinates is called as the seed vigor. Seed vigor index is calculated by multiplying germination and seedling length. Seed vigor index is calculated by multiplying germination and seedling length. The seeds having higher seed vigor index is considered more vigorous. Some of the seeds you put it in water, by next day morning they will germinate. While some of them you keep for 1 day or 2 days, then they germinate. So automatically one which germinates in few hours or less time, they are considered to be more vigorous than those which will take 2 days, 3 days to germinate. Okay, therefore seed vigor index is one of the important parameter which decides the rate at which the seed will germinate. Okay, so if the seed germinates very fast, we say that the index is very high, and if it germinates slowly, we call it as the vigor index is low. So this is about seed vigor index. Okay, thank you very much. In this lecture we have studied till this part only.